Hello, in this video we're going to be looking at what is DNA. So let's not waste any of your time. Let's get right to it.
It's hard to breathe, but that's alright. Hush.
Now, when we're talking about what is DNA, what we're going to do is kind of take a historical look at the series of scientists that got us to the point of our understanding about DNA. And of course, in this day and time, the knowledge base of DNA is exponentially growing at all times. After the scientists that we take a look at just in the last 20, 30 years, 40 years, if you go back to 1980, with just the introduction of technology and the improvement in technology, what we can do now with our understanding of DNA is just way out there, even to the point of, the process of the 90s and the early 2000s, when they completely broke down each gene to figure out what trait it's determined for. So that's what we're going to be doing. We're going to take a look at these 6. Actually, it's 7. I forgot to include one in my list there. I made the list, and then I found another one that I wanted to include. But we're going to be taking a look at these experiments that these scientists conducted and what their contribution was to our understanding of what is DNA. And we're going to look at from the standpoint of who they are, what they discovered, how they did their experiments, because that's really the interesting part of all that, and of course why it's important in their conclusions. Now, when we want to think about what is DNA, some little background information. The very first time that a scientist pointed at something and said, "Hey, that's nucleic acid," that was Frederick Michener, as you can see on your screen, 1869. Found him studying skin disorders and that kind of stuff. But really, the two main functions that nucleic acids have, and it's going to be part of when we talk about replication. It's the background of what we're going to be thinking about. It's the fact that one, that it stores and contains genetic material. All the genetic material is going to be passed on from parent to offspring, the traits. We talked about in the last video series the idea of heredity and passing that on and how the traits are blended and stuff, and through meiosis and things like that. But number 2, the storage of the information, with the idea of it has the instructions for making the proteins. When it gets translated to RNA and then the proteins are made, those instructions, those blueprints that are there. Okay, man, I shouldn't say we all, but most of us, I'm sure, love those police documentary type series. Not, I shouldn't have said documentary, where there's a police force and they're solving crimes, and of course it seems to be about a murder, and then being able to find clues at the crime scenes, and one of those things that they always are looking for is DNA. They can take that DNA. Of course, we know they can match it up to an individual person, and that's always the goal. The forensic team, they swoop in and just scrub everything, just trying to find that golden piece of evidence. But when you're thinking DNA, it's that DNA chromosomes that they're looking for. It's the same idea with matching an offspring to a parent. to determine, if that's most cases, if that's the dad or not. So, but it's just it's a fascinating part of those types of shows. All right, so here's our first scientist, Frederick Griffin, as you can see there. Uh, his contribution to this whole process of learning what DNA is the fact that in his experiments, and I should have led with this earlier, that I'm going to try to, a lot of these scientists, they have multiple slides and multiple steps in their experiments. And on some scientists, you're going to see the same diagram on several slides in a row, mainly because it's a diagram about their experiment, but their experiment has many steps, and just to make sure that the text that I put on there for you to take notes on isn't so jam-packed and so small that it's not legible. The steps have to be broken out into a series of slides. So even though you may see the same diagram, the text that's on there will be different, but you'll notice that. And I will try to, with each slide, talk about the steps of the written text on there. Hopefully I'll pay attention enough to be able to, I won't tell the whole story, and then we got 2 or 3 slides where there's no more story to tell, but there's information to be had. So we're going to try that. But our first scientist, Frederick Griffin, his contribution to this whole gamut of trying to determine and understand what DNA is that he found that with bacteria, okay, something could be exchanged. One bacteria could give something to another bacteria to kind of alter what it does. And what we mean by that is he looked at, you can see there, Streptococcus pneumonia. It has two strains, a one that was dangerous and deadly virulent, the smooth, and then the rough, which was non-virulent. In other words, if you had the rough, it wasn't going to affect you, but if you had the smooth, that could affect you greatly. Okay, so again, he had the two types, the S, which was disease causing, the R, which was not. And what he found is that he took to start off the process. He took some of the S and injected with mice, some of the R injected with mice, and the mice that were injected with the S strain died. The ones that were injected with the R strain did not die. Let's see. I think I've already. Yeah, see, I'm already getting ahead of myself. I need to slow down on my steps. But the idea of the S strain killing the mice, the R strain not killing the mice. But then what he found is that when he took the R strain and the S strain and put them together, okay, into one container, and then injected that into the mice, the mice died, much to his surprise. And when he went to he investigated the mice, studied the mice, and pulled strains of bacteria, the Streptococcus, out, he did not find any R's. And so what was happening is that the S's were somehow transforming the R's. They were somehow passing something that made made them into virulent bacteria. So that was the first discovery, was that something could be passed between bacterias that could change the other one. And you think about that, he looked at, the pneumonia disease could be transferred I don't want to give away what yet, but it could be many things could be transferred in this way. It could be, DNA. You could, if you were a bacteria that showed immunity to a particular antibacterial medicine, and you survived, you could pass that on to others, and then they would become antivirus. So there's a lot of things that could be passed on between the bacteria. Okay, so our next, I need to go back and say this information I'm going to share should be back with Griffith. It's the idea of bacterial transformation. That's the process, and it was on one of the screens, so if you've written everything down, you've got this. But it's the idea of being able to transfer DNA from one bacteria to the next. And research companies use that even today to make in the idea of farming and seed production. They want to make seeds that are going to produce a healthy plant, whether it's corn or soybean or whatever. So they've taken DNA from bacteria that show signs of things that would be helpful for the plant, like somehow inhibit insects from eating the leaves and stuff, or growth factors, that kind of thing. So there are applications that are used today, which is the same idea, and instilling DNA into one thing from something else. Excuse me. So it's that idea. So our next scientist, Oswald Avery, with Griffith, he determined that something was being passed, but did not know what. Avery came along and conducted experiments, again with the smooth virulent, except he wanted to decide, okay, what actually is being passed from bacteria to bacteria. So he did an experiment and determined that it was DNA that was being transformed. All right. So what Avery did, in essence, was to take a solution with the S strain bacteria in it, and put that into the solution. And of course, he used RNA, he used DNA, and he used just proteins to see what the S strain would affect. And then he used enzymes that would destroy each of those three. And he simply just would take, getting ahead of myself again here. Okay, simply took the solutions with the enzymes and added the three components after the S strain had a chance to work on it. And then, of course, took those solutions and injected them into mice to see which ones would still be harmful to the mice. And the solution that had the DNA, after the DNA was broken down, it would no longer be detrimental to the mice, So that was his conclusion, that it was the DNA that was being sent or put into another bacteria from one bacteria, transformed. The idea that it was DNA was the material that was being passed from bacteria to bacteria. All right, so our third scientist, Hershey and Case. Now, this set of team, they reached the same conclusion, that it was DNA. They just did it in a different manner, with a deeper understanding of how this process worked, because they went more from the bacteria side. It's structure based, and that'll make more sense once we see the next slide. But it's the idea that they confirmed, but they did it in a much with a different experiment. Okay, before we get into their experiment, let's just remind ourselves of how bacteria does this. You can see the diagram beside me there. You've got bacteria that will infect a cell, and when that happens, what we're saying is that it's just injecting its DNA into the cell, and then allows the cell's own machinery to reproduce that DNA. Excuse me. That DNA to allow more bacteria to be made. But where this DNA comes from is the phage section, and that's a little capsule that's on top of what looks to be the spider-like body for that. All right, so getting into their experiment, what they did is they took phosphorus and sulfur. Okay, phosphorus is found both in DNA and in proteins. So if they coat that, or if they make it radioactive where they can trace it, that was the goal. And they also used sulfur. So they could eventually see where the bacteria DNA was. That was the point, to try to trace it, to see where it ended up. Again, looking to see exactly what is being transferred. All right, so and here's their experiment. Now you'll see the pictures of the blenders and wonder, what, a blender? Wow, that's not a very scientific tool, and we would not think so. The idea of a centrifuge does the same thing. It spins to try to separate out the different components of whatever it is you're spinning, and a blender obviously spins. So they were using that. But it's the idea of to try to isolate the material being transformed. They had one solution with just the phage capsule only, and then the second one with just the bacteria body itself. And the idea of putting them in the blenders and spinning those, and then being able to separate out the different components by density. And they found the radioactive DNA that could cause diseases in the lower part of the solution. You can see there in the diagram on in the test tube on the left. But that phage capsule sinking to the bottom, and the parts that were in the the watery solution, the supernat fluids, and of course, and you can see there on the diagram that that section at the bottom, or what's in the bottom, is called a pellet. That's where it found the radioactive phosphorus DNA. So again, confirming that it's the DNA. Alright, so again, the idea of just using that blender to separate it, you spin it, and the phage part will come separate from the bacteria, and the idea of being able to trace what's going on. And again, he found it in what was called the pellet. So the pellet's the word you're going to want to look for if you see a multiple choice question on this topic. And so again, just the conclusion one more time. It was the phage DNA that is being sent to another, or could be sent to another bacteria. So the idea of that DNA being super important, because in this situation it can be transferred to something else. Well, that's our first video on the scientists and their experiments. And when we come back to the next time, we'll look at experiments part 2. We'll see you then.